Okay, good afternoon, students. In this lecture, we are going to discuss about the morphology and anatomy of the seeds. Okay, today's topic for the discussion is the morphology and anatomy of seeds. Okay, first of all, what is meant by the seed? In broad sense, when we are talking about the seed, it is the material which is used for planting or the regeneration purpose. Okay, in the broad sense, seed is a material which is used for planting or regeneration purpose. Scientifically speaking, seed it is a fertilized ripened ovule consisting of three main parts, namely seed coat, endosperm, and embryo, which in due course give rise to the new plant. Okay, when scientifically speaking, the seed it is a fertilized ripened ovule consisting of three main parts, namely seed coat, endosperm, and embryo, which in due course gives rise to the new plant. Okay, then the seed it is also referring to the propagating materials of the healthy seedlings. Okay, any propagating material of healthy seedlings, tubers, bulbs, rhizome, roots, cuttings, cutting sets, then all types of grafts and vegetatively propagating materials used for the production purpose. All these are referred as the seed. Okay, in the broad sense, the seed it is a material which is used for planting or the regeneration purpose. Scientifically, seed is a fertilized ripened ovule consisting of three main parts, namely seed coat, endosperm, and embryo, which in due course gives rise to the new plant. Okay, then the seed it is also refers to the propagating materials of the healthy seedlings, tubers, bulbs, rhizome, roots, cutting sets, all types of grafts, and the vegetatively propagating materials used for the production purpose. Okay. Then there are two types of the seeds. All you know that there are two types of the seeds. The first one is the monocot seed, and the another one is the dicot seed. Okay, what is the difference between these monocots and the dicots? Or we are saying that what is the difference between these two types of the seeds? Okay, here you can see the monocots are these are the plants in which the seeds are having the embryo with the single cotyledon. Okay, mono means single, monocots as the name suggests, that means the embryo it is having with the only single cotyledon, while in the dicots the seeds are having the embryo which are with the two cotyledons. Okay, embryo with the two cotyledons, such type of the seeds are referred as the dicots. Di means two, as the name suggests, the embryo with the two cotyledons are referred as the dicots. Okay, then the monocots, these are usually endospermic seeds. Okay, endospermic seed means what? Here that is the food material, it is stored in the form of endosperm. Okay, and that's why the monocot seed these are usually endospermic seeds. In other word, the endosperm these are endospermic seeds, these are also referred as the albuminous seeds. Okay, these are also referred as the albuminous seeds. Endosperm, that is it is having the food material which is stored and which is utilized in the germination of the seeds. Okay, these are the monocots which are usually endospermic. Okay, while the dicots these are usually non-endospermic. Okay, here the endosperm it is not present and the food material it is stored in the cotyledon. Okay, these are usually non-endospermic, but there is one exception. For example, the castor seeds, these are the dicot seeds, but these are having the endosperm. Okay, non-endospermic seeds these are also referred as the non-albuminous seeds. Okay, dicot seeds these are usually non-endospermics except the castor seeds. Okay, castor seeds these are dicot seeds, but these are having the endosperm. Okay, then the germination usually hypogeal, that is except in the onion. Okay, germination usually hypogeal means what? Here hypogeal, that means the two cotton during the germination, that is they come above the surface of the soil. and that's why such type of the germination, it is referred as the hypogeal type of the germination, and which is very common in the monocot except in the onion. While in the dicot the germination usually epigeal, that is epigeal, except in the p and the p gram. Other dicots, these are epigeal. Epigeal type of germination means what here? That is the two cotyledons, they never come on the surface of the soil, the germination process, and such type of the germination is called as the epigeal type of the germination. Okay, in the dicot the germination is usually epigeal except in the p and the gram. Okay, here that is the plants in which the monocot seeds are produced. Generally the flower parts in the multiples of 3 flower pots. Flower parts means the calyx, corolla, androecium, and the gynoecium. Generally, these floral whorls, these are the floral parts. These are in multiples of 3. While the plants which are having the dicot seeds, these plants in which the flower parts, these are in multiples of 4 or the 5. Okay, flower parts in the dicotyledonous plants, these are in multiples of the 4 or the 5. Then here all you know that in the monocot plants the major leaf veins are parallel. Parallel leaf veination, it is seen, it is observed in the monocots, while in the dicot plants the leaf veins, these are reticulated. Okay, reticulated variation, it is observed on the leaf in the dicot plants. Then the stem vascular bundles are scattered in the monocot plants. Okay, stem vascular bundles, these are scattered in the monocot plants, while in the dicot stem vascular bundles, these are present in a ring. Then the root system, it is adventitious in the monocot plant, while the roots, these are developing from the radicle. And generally the root system in the dicot plant, it is a taproot system. Okay, the root system in the monocot, that is the adventitious type of root system is present, and in the dicot the taproot system is generally present. Okay, then in the monocot the secondary growth is entirely absent, while in the dicot the secondary growth is often present. Okay, monocot secondary growth is absent, but in the dicot the secondary growth is often present. Okay, here you can see the examples of the monocot, the grains like the wheat, corn, rice, millets. Okay, then the sugarcane, banana, ginger, onions, and the grasses, these are the monocotyledonous plants. While the legumes, for example, pea, beans, lentils, peanuts, then mint, then lettuce, tomato, oak, tree, that is all these are dicotyledonous plants. Okay, then the structure of the dicot seeds, we are going to discuss the, just we have discussed the differences between the dicot plant and the monocot plant, and now we are going to discuss about the structure of the dicot seed and the monocot seeds. Okay, there are certain differences between the monocot and the dicot seed. The first of all, we are going to discuss the structure of the dicot seed. Okay, here the dicotyledonous seed, as the name suggests, has the two cotyledons. Okay, di means the two, and that's why it is having the two cotyledons, and it has the following parts. Okay, the first most important part, it is the seed coat. Okay, seed coat, this is the outermost covering of the seed. The seed coat has the two layers. The outer layer, it is called as the testa, and the inner layer, it is called as the tegmen. Okay, the seed coat, seed, it is covered by the layering, and that layering is called as the seed coat. And generally in the dicotyledonous plant, the seed coat, it is two layered. The outer layer, it is called as the testa, while the inner one, it is called as the tegmen. Okay, then another part which is present, it is called as the hilum. Hilum, that is the, it is the scur on the seed coat through which the developing seed was attached to the fruit. Okay, the hilum, it is the scur like marking which is present on the seed coat through which the developing seed was attached to the fruit. Okay, then the micropyle. Micropyle, it is the small pore present above the hilum. Okay, micropyle, it is the small pore which is present above the hilum. Then the most important part of the seed, that is the living part of the seed, it is referred as the embryo. Embryo, it is consist of an embryonal axis, and it is having the two cotyledons. Okay, it is the living part of the seed, and the embryo, it consists of the embryonal axis and the two cotyledons. Okay, the seed coat, the outermost covering, which is two layered, outer one is called as the testa and the inner one is called as the tegmen. The hilum, it is the scur like structure which is present on the seed, with which the seed was attached to the fruit. Then the micropyle, it is the small pore which is present above the hilum, and the embryo, that is the living part of the seed, which consists of an embryonal axis and the two cotyledons. Okay, here we can see the two cotyledons. Cotyledons, these are often fleshy and full of reserve food material. Okay, cotyledons, these are often fleshy and full of reserve food material. Okay, then the radicle and the plumule, just we have discussed that is the seed, it is having the embryonal axis, and this embryonal axis having the two ends. The one end is called as the radicle, and the another one is called as the plumule. Okay, here the radicle is the structure which further undergo the developmental stages and then get developed into the root system after the germination. Okay, after the germination of the seed, the radicle and plumule, it comes out of the seed. Then the further development of radicle takes place, and the radicle, it will get developed into the root system, while the plumule, it get further developed into the shoot system of the plant. Okay, radicle and the plumule, they are present at the two ends of the embryonal axis. Then the endosperm. Okay, just we have discussed that the most of the dicot seeds, that is these are non-endospermic. Okay, in some seeds, such as castor, it is a dicot seed, but in the castor, the endosperm, it is formed as a result of double fertilization, and it is a food storing tissue. Okay, endosperm, it is nothing but the food storing tissue. Generally in the dicot plants, the food, it is stored in the two cotyledons and the endosperm is absent. That means the dicot plants, mostly these are non-endospermic, but there is only one exception, and that is the castor plant, in which the seeds are endospermic. Okay, endosperm, it is a food storing tissue. Okay, in plants such as the bean, gram, and the pea, the endosperm is not present in the matured seed. They are known as the non-endospermous. Okay, these are known as the non-endospermous. Okay, you can see the in the first photograph, you can see the structure of the gram seed. Okay, here you can see the testa, then here you can see clearly the two cotyledons of the plant, and then it is the seed coat. The seed coat, it is made up of two layers. The outer one, here you can see, it is called as the testa, while the inner one, it is called as the tegmen. Okay, then here the embryo, it is having the two axis. The embryo, it is embryonal axis and the two cotyledons. In these two cotyledons, the abundant food material, it is stored, and in the gram seed, the endosperm is entirely absent. That means the gram seeds, these are non-endospermic. Okay, and here the food material, it is stored in the two cotyledons. Then the embryonal axis, it is having the two ends. Here you can see the one end, it is referred as the radicle, and another one, here you can see the plumule. The radicle further get developed into the root system, and the plumule, it further get developed into the shoot system. Okay, here in this one, it is the opened gram seed, and here it is, you can see the hilum. Hilum, it is the scur with the by which, that is the seed, it is attached to the fruit. Okay, then the micropyle, that is the opening of the hilum, here you can see. Okay, that is the structure of the gram seed. Gram seed, it is a non-endospermic. Okay, but here you can see the castor beans. Castor, it is dicot seed, but though it is a dicot seed, it is a endospermic seed. Okay, here you can see the seed coat is present. Here the seed coat is present. Then here you can see this is the endosperm, that is the food storing tissue is present. And here you can see the two cotyledons, presence of the two cotyledons, and here the radicle which further get developed into the root system. Okay, that is mostly the dicot seeds. These are non-endospermic. Food is stored into the two cotyledons, but the castor, it is the exception in the dicot plants, because it is having the two cotyledons, and along with this it is having the food storing tissue called as the endosperm. That means the castor seeds are endospermic. Okay, that is all about the structure of the dicot seed. Okay, then the structure of the monocotyledonous seed. Structure of the monocotyledonous seed. A monocotyledonous seed, as the name suggests, has only one cotyledon. There is only one outer layering of the seed coat. Okay, just we have discussed in the dicot, the seed, it is covered by the seed coat, and the seed coat, it is made up of two layers. The outer one is called as the testa, while the inner one is called as the tegment. But in the monocotyledonous seed coat, in the seeds of the cereals such as the maize, the seed coat, it is membranous and generally fused with the fruit wall called as the hull. Okay, that is the particular character, that is a very specific characters in the monocotyledonous seed like the maize, where the seed coat, it is firmly attached, firmly fused with the fruit wall, and it is called as the hull. Okay, then the endosperm, monocotyledonous seeds, that is, these are generally endospermic seeds. That means the food storing tissue is present. Endosperm, the endosperm is bulky and stores the food. Generally the monocotyledonous seeds are endospermic, but some, as in orchids, these are non-endospermic. Okay, mostly the monocotyledonous seeds are endospermic. Endosperm, it is the food storing tissue. Okay, then the another important covering of the endosperm, that is called as the alluron layer. Okay, alluron layer, that is the outer covering. The endosperm, it is covered with the alluron layer, the outer covering of endosperm, which separates the embryo by the proteinaceous layer called as the alluron layer. Alluron layer, alluron, it is a type of the protein. Okay, that is, these are proteins, these are covering the endosperm, and thus differentiating the endosperm from the embryonal part. And that's why this layer, it is called as the alluron layer. Alluron, it is a type of the protein which is separating the endosperm from the embryo. Okay, and that's why that particular layer is called as the alluron layer. Okay, then the presence of the embryo, that is the important part of the seed, that is the living part of the seed, embryo. The embryo is small and situated in a groove at one end of the endosperm. Okay, the embryo is small and situated in a groove at one end of the endosperm. Then scutellum, scutellum, large shield shaped cotyledons. Okay, here scutellum means what? Here the um, in the monocotyledonous plants, only one cotyledon is present, and that large and shield shaped cotyledon is referred as the scutellum in the monocotyledonous seeds. Okay, scutellum means, that is, these are the large shield shaped cotyledons. Okay, then the embryonal axis, you know, embryonal axis, it is the one end is called as the radicle, and the another one is called as the plumule. The radicle further get developed into the root system, while the plumule get further developed into the shoot system. Okay, then the coleoptile and the coleorhiza. Okay, here the embryonal axis, plumule and the radicle, these are covered with the special type of the sheath. Okay, the plumule and radicals, these are enclosed in the sheath, and these are called as the coleoptile, the coleorhiza. Okay, we can say the protective sheath of the plumule and the radicle is referred as the coleoptile and the coleorhiza. Okay, that is, this can be very clear by observing this photograph. Here you can see it is the ELS of, or the, we can say, the longitudinal section of the maize grain. Okay, here you can see the here the seed coat and the fruit wall, these are firmly attached with each other. Okay, here you can see the fused pericarp and the testa, and that's why this layering, the seed coat, it is referred as the, in the mm, maize grain. Okay, it is referred as the maize grain. That is the important characteristic feature of the monocot seed. Okay, then here you can see the endosperm is present. Okay, here you can see the endosperm, that is the food storing tissue is present, where the food it is stored in the form of starch. Okay, it is the storage region, or it is called as the endosperm. And that endosperm, it is then covered with the alluron protein. Here you can see this is the covering of the alluron protein, and that's why this layer it is referred as the alluron region. Okay, this layer is called as the alluron region. Then here you can see the cotyledon, a shield like structure is present, that is the cotyledon, and this cotyledon it is called as the scutellum. Okay, it is called as the scutellum. Then here you can see this endosperm, it is separated, it is differentiated from the scutellum by the presence of the layer, and this layer is called as the epithelial layer. Okay, the endosperm and the scutellum, cotyledon, it is differentiated from each other by the presence of a layer, and that layer it is called as the epithelial layer. Okay, then here this is the shield shaped cotyledon, which is referred as the scutellum, and at one end, or at the one corner of that scutellum, there is a presence of the embryonal axis. Okay, here you can see this is the embryo living part of the seed which is present, and here the one end, one pole of that embryonal axis is called as the radicle, which further get developed into the root system. And here this radicle it is covered by the specialized sheath, and the covering or the sheath of the radicle it is called as the coleorhiza. Okay, the covering of the radicle it is called as the coleorhiza. And here the one pole, or the one end of the embryonal axis, it is the embryonal axis, one end is called as the radicle here, and the another pole, another end of the embryonal axis is called as the plumule. Plumule further get developed into the shoot system. Here plumule also it is well protected by the covering or a sheath, and that sheath or the protective covering of the plumule it is called as the coleoptile. Okay, it is called as the coleoptile. Okay, that is all about the morphology and the anatomy of the seeds.